Welcome to today's lecture on the cell biology of the digestive system. We will explore the complex processes that govern digestion, including the role of intestinal stem cells in tissue regeneration and the interactions between gut microbiota and the host. Let's dive into this fascinating topic together. Within the lining of the intestines, we find a remarkable population of cells known as intestinal stem cells. These specialized cells possess the unique ability to self-renew and differentiate into various cell types, which replenish and maintain the intestinal tissue. When injury occurs, such as damage from inflammation or infection, these intestinal stem cells actively proliferate and differentiate to replace damaged cells and restore tissue functionality. Their remarkable regenerative capacity ensures the maintenance of a healthy intestinal epithelium. In the video on cell biology of the digestive system, we will focus on the importance of the intestinal mucosa and the gut-associated lymphoid tissue, GALT, in immune protection against pathogens and maintaining balance with the gut microbiota. Effective communication between cells is vital for the coordination of various digestive processes. In the gut, cell-cell communication occurs through various mechanisms, including direct cell-to-cell contact and the secretion of signaling molecules. Tight junctions between epithelial cells form a physical barrier preventing molecules from freely passing through the cellular layers. However, specialized transporters and channels allow necessary nutrients to be selectively transported across the intestinal epithelium. Additionally, paracrine and endocrine signals, such as hormones and neuropeptides, regulate digestive processes by acting on specific receptors present on target cells. The small intestine is responsible for the absorption of nutrients from digested food. The lining of the small intestine is highly specialized, with finger-like protrusions called villi and even smaller microvilli. These structures increase the surface area available for nutrient absorption. Epithelial cells lining the villi possess transporters and channels that facilitate the uptake of nutrients, such as amino acids, sugars, and lipids, into the cells. Once inside the cells, these nutrients are either transferred directly to the bloodstream or processed further before being released into the circulation. The efficient breakdown of food components relies on the production and secretion of digestive enzymes. Various cells in the digestive system, including specialized cells in the stomach and pancreas, synthesize and release these enzymes. Hormonal signals, such as gastrin and secretin, regulate the secretion of enzymes from these cells. Gastrin stimulates the secretion of gastric enzymes, while secretin activates the pancreas to release enzymes involved in the digestion of carbohydrates, proteins, and fats. These synchronized actions ensure optimal food breakdown and nutrient extraction. The development and differentiation of digestive system cells are tightly regulated by a complex network of molecular pathways. Signaling molecules, such as growth factors and morphogens, guide the proliferation and differentiation of precursor cells into specific cell types. For example, the WNT signaling pathway plays a crucial role in intestinal stem cell maintenance and differentiation. Perturbations in these signaling pathways can lead to developmental defects or abnormal cell differentiation, resulting in digestive disorders. The gut microbiota, composed of trillions of microorganisms residing in the intestinal tract, significantly influences digestive system homeostasis. These diverse microbial communities interact with intestinal cells, modulating various physiological processes. They aid in the breakdown of complex carbohydrates, produce vitamins, and contribute to the maturation of the immune system. Imbalances in the gut microbiota composition, known as dysbiosis, have been associated with gastrointestinal diseases, highlighting the importance of maintaining a healthy microbial community. The enteric nervous system, ENS, is often referred to as the second brain due to its extensive neuronal network lining the entire gastrointestinal tract. The ENS regulates peristalsis, the rhythmic contraction of intestinal muscles that propels food along the digestive tract. Neurons within the ENS receive signals from the central nervous system and respond to local environmental cues, coordinating digestive processes. Disruptions in ENS development or function can lead to motility disorders, such as irritable bowel syndrome. Complex carbohydrates, such as dietary fibers, serve as an essential energy source for the gut microbiota. However, the human digestive system lacks the enzymes required to degrade these complex carbohydrates. Instead, specialized microbial enzymes produced by gut bacteria break down complex carbohydrates into simpler compounds, such as short-chain fatty acids. These compounds are then utilized by intestinal cells, providing a critical energy source for their functioning. The intestinal epithelial barrier functions as a selective barrier, allowing the absorption of nutrients while preventing the entry of harmful pathogens and toxins. Tight junctions between epithelial cells form this barrier, maintaining the integrity of the intestinal lining. Various factors, including cytokines and signaling molecules, actively regulate the tight junctions and permeability of the epithelial barrier. Dysfunction in these processes can lead to increased intestinal permeability and the translocation of harmful substances, contributing to inflammatory bowel diseases. The stomach and small intestine encounter varying levels of acidity during the digestion process. To adapt to these acidic environments, specialized cells within these regions possess unique adaptations. In the stomach, for instance, parietal cells secrete hydrochloric acid, creating an acidic environment necessary for protein digestion and the activation of digestive enzymes. In the small intestine, the secretion of bicarbonate ions by the pancreas and the release of mucus by goblet cells protect the epithelium from acidity, ensuring proper digestion and absorption. Digestion and appetite regulation involve a delicate interplay of hormonal signaling and neuropeptides. Key hormones, such as ghrelin, leptin, and peptide G, are involved in signaling hunger and satiety to the brain, influencing food intake. Additionally, neuropeptides, such as neuropeptide Y and alpha-melanocyte stimulating hormone, modulate feeding behavior and energy expenditure. Dysregulation of these signaling pathways can contribute to metabolic disorders, such as obesity and eating disorders. In summary, the cellular processes in the digestive system are responsible for breaking down food, absorbing nutrients, regenerating tissue, and triggering immune responses. From stem cells to gut microbiota, these topics highlight the complex interactions within the digestive system. Understanding these mechanisms is important for diagnosing and treating digestive disorders. Ongoing research continually reveals new findings in cell biology within the digestive system.